transcription is, remember, going to be part one of protein synthesis. So we do have to keep in mind that when we're looking at the process of transcription, it's part of the overall goal of protein synthesis. So one of the things that we really need to be able to understand in order to understand transcription is the structure or architecture of a gene. So keeping in mind that DNA, the whole genome, is going to contain lots of small units called a gene. That gene is really going to have three main parts. It's going to have the promoter, the coding region, and then a terminator. So we do need to know these kind of three words: promoter, coding region, and terminator. So the promoter is going to be what we call upstream of the gene, and this is going to be where transcription factors are going to assemble, and that's going to attract the RNA polymerase. So when we talk about this main step, initiation, we're really keeping in mind that whatever protein needs to be made, certain signaling factors are going to take us to that gene. So a lot of these transcription factors are going to really be signals, some sort of signal that tells the cell we need that gene. A very common component, just a small part of a promoter, is usually called the TATA box. And the TATA is a region that begins with TA. Keeping in mind between A's and T's, there's only two hydrogen bonds. So this region will be a little bit easier to unzip. So we do want to recognize the TATA box, and then the fact that we're usually going to transcribe after the TATA box. So the promoter is not transcribed. During elongation, we do want to keep in mind that we are synthesizing mRNA, and we're still building in the 5' to 3' direction, and so that would mean that we're actually building on a 3' to 5' template. So an interesting thing here is that at this particular moment we're only using one side of the double helix, and that's the template strand. The coding strand is what we call the upper strand, because when we do transcription, remember, we're not copying. We're really using base pairs to do this building. So for example, if the DNA sequence on the template strand were ATC, AAA, we would of course be able to figure out the other side of the DNA. The other side of the DNA would be TAG, TTT. And there's our DNA sequence. If we wanted to transcribe, then we know we use RNA base pairs. This is why we really needed to know the structure of RNA. And so then our RNA would actually be U, A, G, U, U, U. And this is transcription. It's using a template to attract base pairs. We still have an enzyme, RNA polymerase, and it's going to bring the appropriate nucleotide. Keeping in mind when we write this, all that we're really writing are the nitrogen bases, but a sugar phosphate backbone will be formed as well. Again, RNA nucleotides. And then once we reach the terminator, we get to a sequence called the termination phase. So we have initiation, elongation, termination, and RNA polymerase is going to be released. So the enzyme is released. So this is still something they're really looking at, but something in this sequence down here releases or does not allow the enzyme to continue. And so when RNA polymerase is released, the DNA re-zips, or we reform our DNA base pairs, and the mRNA is also released. So our end result is that we have a complementary sequence of a gene that can travel to the ribosome. So the base pairing is one skill that you need, but you also really need to know the architecture of this. The concept of a promoter, a coding region, and a terminator. The concept that when we unzip or break the hydrogen bonds, that we actually have two strands. One is the template that we build on. The other is called the coding strand. Because if you look at the relationship, we do see some similarities. Do not fall into the trap of trying to just change T's to U's. Because you don't know if they're going to give you the template strand or the coding strand. So you need to just be able to use your base pair rules. And so this is kind of the end of transcription. The DNA will close back up and the mRNA will go to the ribosome. However, they have discovered that in eukaryotic cells, there is another step, and it's called post-transcriptional modifications. So at the end of this, what we actually have is an mRNA sequence. But in eukaryotes, it's called a pre-mRNA sequence because we are going to do some modifications. So here's our architecture of our gene, and it turns out that the mRNA that we want needs to have some areas removed. And so we have these words introns and exons. What I want you to think of for exon is expressed. And what I want you to think of for introns is interfering. There are actually other meanings behind the introns and the exons, but if you can memorize these words, it will help you remember which ones we actually want to keep. So it turns out that the cell is going to remove some regions. And those regions are called introns. And so those regions are going to be removed. And they are removed by a mechanism called a spliceosome. So a spliceosome is a multi-component enzyme that removes introns and links exons. And so the intron that is removed is going to just be degraded and those nucleotides reused. The spliceosome can do its job over and over again. And so they're also called SNRPs. It means the same thing, spliceosome or SNRP. And so they're going to be part of processing the mRNA. There's a lot of theories about why this is present. One of the theories is that over evolution there were mutations, and so those mutations were removed. However, we'll also see that this is really important for antibody formation. It allows us to have something called alternative splicing. So one advantage that they've seen is that sometimes it's not always the same introns that are taken out. Depending on the needs of the cell, it might take this out, it might take that out, it might take them both out, it might take none out, and that's what we call alternative splicing. So it turns out that even from the same gene, we can get very different mRNAs. And this actually helps create a larger variety of mRNAs, which really means that we'll have a larger variety of proteins as well. So, key point is this is only in eukaryotes. This is after transcription. The mechanism is called a spliceosome or a SNRP, and it's going to help remove the intron and splice or combine the exons together. And then we call it a mature mRNA. Notice that the mRNA actually has an orientation as well. It has a 5' to 3' orientation also. There's one other thing that you'll notice in this diagram, and it's that mRNA will have what we call a cap and a tail. And so we just want to talk about the cap and the tail. This is also only found in eukaryotes. So the process of transcription is the same in prokaryotes and eukaryotes, with the exception that eukaryotes have this step as well, this modification step. The 5' cap is going to be a series of nucleotides added to the 5' end. And the poly-A tail is also going to be a series of nucleotides, but adenines on the 3' end. So what these are basically going to be cushions that are going to help prevent degradation. So keep in mind, as soon as we leave the nucleus, we're going to be in the cytoplasm. There are lots of enzymes, there are lysosomes, there are lots of things that could damage the mRNA. So putting this cap and tail onto it is going to help protect it as well. It also helps signal for exiting the nucleus. So remember, the nucleus is very selectively permeable, only allows certain things to move across the membrane, much more so than the cell membrane. So the nuclear membrane is very selective. So we need a signal to help us exit the nucleus. We also need a signal to bind to the ribosome. So they believe that the cap and the tail are going to help with that, as well as prevent degradation. So taking a second to stop and think, do I know what transcription is? This is where you really do need to know the differences between DNA and RNA. You'll want to be able to kind of predict what would happen if a promoter region were damaged, if a signal weren't received. You want to be able to explain how transcription can actually happen. And then we know this part about the caps and the tails and the introns and the exons. We keep in mind this is part of the overall process of protein synthesis, but that at the end of transcription, we still are going to have to go on to translation. So even though we now have an mRNA sequence, we are going to need to begin the next stage. There are lots of different animations and tutorials that you can practice with. And so you want to know what a gene is, a sequence of DNA that codes for a protein. You want to know the types of RNA, the structures of DNA and RNA, the steps of transcription, those three main steps. You do want to take a second and compare and contrast DNA replication with transcription. There are some things that they have in common, but there are some things that are very different. So you want to make sure that you can separate those in your brain. Describe the different results of post-transcriptional modification, and then talk about Beadle and Tatum's definition of a gene, one enzyme, and compare that to what we get, especially keeping in mind those post-transcriptional modifications. And that really is transcription. From here, you just really have to do some practice.